So as mentioned in this webinar, we would like to introduce you to the European Lead Factory, which is a public-private partnership funded by the Innovative Medicines Initiative. We have been set up to boost early drug discovery, mainly in Europe. In this webinar, we would like to explain you the concept of the European Lead Factory, how you can benefit from a collaboration. We have an example of what a collaboration with the ELF can lead to. And we also have a presentation on the public compound collection. We will start off with a talk by Jon de Vlieger, who already has his camera on, as I can see. But for later today, we have presentations from Dr. Raymond Pierce, who's an Emeritus Director of the CNRS, and a talk by Dr. Hugues Lemoine, who's a chef de unité of the chemical library of Edelris. So towards the end of all these, and towards the end of the whole session, we will also have a Q&A with experts from the European Lead Factory. So you can ask your questions, any specific questions that you might have on assay development, for example, or high throughput screening, high content screening, chemistry. So we'll also have that on the menu for today. But before we open, I'd first like to briefly hand over to Professor Rebecca Deprez, who is the president of the French Medicinal Chemistry Society, for a brief introduction and a welcome on behalf of the SCT. Rebecca, the floor is yours.
Vera, I've just had word from Rebecca that she's having some trouble connecting, unfortunately. So we're trying to sort that out, but perhaps we should move on for time's sake.
That's okay. Then I suggest we immediately dive into the introduction to the European Lead Factory from Jon. And after that, we hope Rebecca is able to connect, and we can also, then we'll make a little bit of time for her to announce to talk to the audience. It's okay. I'll take down my slides, and then Jon, the floor is yours.
Wonderful. Thank you so much, Vera. Let me share my screen. Presentation that I'd like to use for today. My name is Jon de Vlieger. I'm the coordinator of the European Lead Factory. You should be seeing my screen by now. Vera, please confirm.
Yes, it's working.
Wonderful. What I will be doing in the next 15 to 20 minutes is to give you a brief introduction to the European Lead Factory. But before doing so, I do see that Rebecca Deprez has joined us. So Rebecca, may I give you the floor for some welcoming words on behalf of SCT?
Yes, can you hear me?
Perfect.
Oh, great. I had problems of connection, so thank you, Jon. Maybe I could have my slides.
Yeah, I'll put your slides up.
Thank you, Vera. So thank you very much. So welcome, everyone. I'm gonna do it in English for everyone. As you've seen, the program will be either in French or in English for everybody to express easily and exchange easily. You can choose any of these two languages. So this is the first Société de Chimie Thérapeutique webinar. So we are really happy and grateful to your European Lead Factory for setting this up with us. People at Lygature will explain to you how European Lead Factory can be a great asset in your project. And the program has been set up with a mix of presentations and experience sharing and discussions. So feel free to intervene and ask a question whenever you want. I would like first to thank especially Brigitte Lesure from the SCT. She's our vice president, and she took this project on behalf of the SCT to set up everything with great people at Lygature, especially Vera, who's the co-host today. And I'm really grateful for their work. I'm really happy also to attend this great program they've put together. Next slide, please, Vera. So a few words about SCT. So as you may know, SCT is an association of medicinal chemists, both from academic and industry and biotech background. So our goal is to promote medicinal chemistry and chemical biology in France and also abroad, as you may know. And for that, we have several actions, such as delivering prizes and awards to medicinal chemistry and chemical biology people, and to organize meetings to support the young researcher in our community. From this year onwards, we will do some webinars from time to time. So this is the first edition, and I'm sure it will be a great success. And we also do some scientific coaching. So if you want to have more information of SCT, please feel free to go to our website, because this is not the main goal today. So I'll go quickly through our actions. Next slide, please. Regarding the events, so we have our very important annual event, which is the RECT in French, or International Conference on Medicinal Chemistry. This year's edition is a fully virtual event, and it takes place next week. So there are still opportunities to register if you wish. In September, we will disclose our new prizes and awards policy and disclose our new sponsors to our prizes. In December, we will have our autumn, sorry for the mistyping, autumn meeting, and it will be dedicated to the search for new antimicrobial agents, and it's co-organized with the French Society for Microbiology. It will be in hybrid mode, both in Lille physically and in virtual on the web. And next February, as every year, we will have our Young Research Fellow meeting, and this year it will be held in Nantes. As for future events, as you know, SCT is a member of the European Federation of Medicinal Chemistry and Chemical Biology. And as the hosting national organization for the EFMC-ISMC next year, we will be involved in the organization of this great meeting that will be held, hopefully physically, in Nice, in France, a great location. And then that's the reason why there will be no RECT next year. And we will go back to the RECT for the 57th edition in Lille in 2023, and then in Bordeaux in 2024. So that's all for my short introduction of this webinar and the SCT. And let Vera, I guess, present Jan, or how, I don't know how, I missed the first minutes, so I don't know.
No problem. I'll hand over directly to Jon.
Thank you so much, Rebecca, for the intro of SCT. I have good memories of the RECT meetings in the past few years. I joined several, and I'm looking forward to meeting the community in real life again.
Yeah.
Also grateful for Brigitte Lesure to link the two initiatives together. From her time at Servier, she has been instrumental to ELF as well. So it's great that the connections really work, also with respect to bringing networks together. So thanks so much. In the next 15 minutes or so, I'll be introducing the European Lead Factory to you. You should be seeing my slide by now. Vera, can you confirm?
Yes, it's working.
Wonderful. As a general introduction to the initiative, and then the real presentations will be given by the different speakers that were announced. So the European Lead Factory is a drug discovery initiative. It is positioned in the early stages of drug discovery, and it typically brings together interesting chemistry and drug targets and phenotypes, which we hope to source from the wider EU scientific community. So when you look at the landscape of drug discovery and development, it's really early stages, bringing together biology and chemistry in hit finding exercises. We are funded through the Innovative Medicines Initiative, the IMI, and that is a public-private partnership between the European Commission and the European Federation of Pharmaceutical Industries. So it's the pharma industry and the European Commission joining each other in this huge initiative to make available the infrastructure and the high quality compound collection for scientific purpose and drug discovery by the wider European community. So we have two main assets in the European Lead Factory. One, of course, is the very unique compound collection. We'll have a detailed presentation on that later in this meeting. But just as a short teaser to that, the collection comprises more than 500,000 compounds, of which around 350 are being put together by the pharma partners, so the industry partners putting in parts of their proprietary compound collections, and around 200,000 of new compounds which were specifically designed and synthesized for this initiative over the past 6 years. So that brings together a nice set of a library for high throughput screening and high content screening that is exclusively available through this initiative. So it's very interesting to have a library, but it's much more interesting to have a library and the infrastructure to screen that library in a high throughput and high content fashion. And that's one of the other assets we have within the European Lead Factory. The full infrastructure to do all the compound logistics, to do the screening, high throughput screening, high content screening, the triaging, and really to bring the best compounds out of this collection that bind, inhibit, activate your specific target of interest. So just a general introduction and frequently used terminology, because I recognize that the ELF, the European Lead Factory, has a specific type of terminology that will come back into this presentation in the next one. So let me go over a few of those. First of all, the term of program owner. And when we are discussing the program owner, we actually mean an academic researcher or a scientist from a small, medium-sized enterprise who has proposed a concept for screening to the European Lead Factory and who is accepted for screening at the ELF. So program owner means someone that has submitted a drug target or phenotype that we have accepted for screening. A program is the screening campaign itself at ELF. So when we are discussing a specific program, it is the full start from really screening, primary screen towards triaging and bringing you the QHL, which brings me to the final term on this slide, the qualified hit list. The qualified hit list is a list of a maximum of 50 compounds, including chemical structures, which are the top compounds from the screening, from the triaging and the resynthesis that are going to be presented to the program owner. So the concept is that we perform all the work, we do the high throughput screening and the triaging, and we present you with a report and the physical samples of the best 50 compounds within our collection that are activating, binding, or inhibiting your target of interest. I briefly mentioned IMI, so the Innovative Medicines Initiative. You will hear the word IMI more often in this call, and FPA, which is the European Federation of Pharmaceutical Industries. So it's the funder, the IMI, and our pharmaceutical partners in the European Commission. MMV is also listed here. This is the Medicines for Malaria venture, and they joined the ELF 3 years ago to use the European Lead Factory for their drug discovery campaigns in malaria research. So what is ELF and what have we been doing up till now? We've been starting activities in 2013, and since then we have been running high throughput screens on over 200 drug targets. So we've been screening the collection of over 500 compounds on over 200 drug targets by now, and that means that we've presented already 7,500 hit compounds and structures to the program owners. And the program owners include academicians from Europe, small, medium-sized enterprises, and the pharma industry participating in this initiative. Just to give you a number of examples of let's say, ELF results that are seen as foundations in the development of new treatments. We have run programs with the German Cancer Research Center, providing excellent starting points for potential new cancer treatments, collaboration with the UK universities on cardiovascular diseases and diabetes, but also with Dutch chemists, Mario van der Stelt, who has progressed the ELF results, so the compounds he obtained from us, and moved that into in vivo animal models. So you see it's a very versatile and diverse group of program owners who have used the ELF facilities and opportunities to bring their programs closer to the next discovery and development phases. So not only academicians have used ELF, also small, medium-sized enterprises. And those small companies, these biotech companies, are also able to secure future funding for the development of their results outside the ELF. So we have seen the announcement of a collaboration agreement between Servier and Lygature, who had obtained interesting compounds from ELF in the field of metabolic diseases, and Servier has closed an agreement with Lygature to develop those compounds. A UK organization, Keepstone Therapeutics, a spin-off from Sheffield University, has secured over €1 million funding from Parkinson's UK to do lead optimization and move their program also closer to clinic. So we also see investment agencies picking up on ELF results. So this is an announcement by an Israeli biotech who has used the ELF infrastructure and library and the opportunity, and is moving closer to clinic with an additional 12.5 million raise from their investors to move things forward. And last example that I'd like to present, and it kind of aligns with what Rebecca just mentioned in the field of antimicrobial resistance. So we have seen Professor Chris Schofield in the UK proposing a very interesting drug target that could potentially lead to new antibiotics. We've screened that, have optimized, let's say, some of those compounds, and then this program was taken up by another IMI program, Enable, and it was very successful, and the team expects to start human trials of these new compounds later this year. So, of course, this is not work that can be finalized within a year. These are very early ELF programs that now are getting closer to clinic, so we can disclose this information to you. So you can see different types of program owners from different countries in Europe, different therapeutic areas, and what they have in common is that the ELF results allowed them to progress their projects and move that closer to patients, either in collaboration with pharmaceutical partners, with investors, with grant funding, or in any other way. So, to be more specific, what are we offering and what is the opportunity for the French research community to work with ELF? First of all, the ELF compound collection being constructed by a part from pharma industry. So 2/3 of the collection is from the different pharma partners participating. You can see all the different partners here. These compounds are not available through any other means and usually are still proprietary. And the new collection, the PCC, which was de novo generated over the past few years, is around 1/3 of this collection. So it's a very diverse collection from different owners, very different chemical spaces, and Hugues will present much more detail also in terms of chemistry later in this call. So let's say we have this library in place and you enjoy all the chemical features that you will see in one of the next presentations. How do we go about screening that collection? Well, first of all, allow me to explain you what we are looking for in a screening proposal. We are looking for innovative screening approaches. And that means that we will have an assessment on the scientific quality of the proposal, the validation of the approach, and also see whether it addresses something new, or perhaps we have already screened your particular target of interest in one of those 200 screens that we've been doing, and then we would really be looking for what it would add to the existing knowledge on this drug target. We would love to see a clear strategy in place how to go about next steps once you receive the results from ELF. What are your plans to, let's say, do hit optimization, lead optimization, etc., and move it to clinic? So we'd like to see the outcomes of ELF really land somewhere with an organization, with a scientist that understands the game of drug discovery and moves it forward. So typically, we have a number of eligibility criteria that we need to check because largely the initiative is public funded. We need to make sure that your institute is eligible for EU funding. So if you're at a French university, a research institute, or a small medium-sized enterprise, this is no issue at all. You are eligible for funding, let's say, the screening program within our framework. Of course, we would love to see that it is different from existing ELF programs. And what is important to mention is that we really need you to have an assay in place. So a screening assay already developed in your facilities that we can transfer into the screening center upon approval of your screening proposal. So how does this work? Let's say your program is accepted for screening. We will be building a program team. You will be part of that team as a program owner, and we will be designing a so-called program plan. And in this plan, we will really list, let's say, the features and the experiments we're going to do and the criteria of which these qualified hit compounds need to meet to be of your satisfaction. So you may be looking for certain selectivity for one target over another. You may be looking for certain chemotypes or not be looking for certain chemotypes. We all agree to those, let's say, criteria. List that down in a program plan, and then once we have agreed, we'll start to work. So we'll transfer the assay into our facilities. We optimize the assay typically towards 1536 when it is high throughput screening, and otherwise 384 if it's high content screening. And we'll perform the work within the facilities of the ELF. So once we have come to, let's say, a top list of compounds, we will go about clearing those compounds for release into your report. The qualified hist compounds is really a maximum of 50, and we will perform on the best compounds of that list a resynthesis to make sure that we have a fresh sample. We retest those fresh compounds so we're sure that the activity seen in the screen is really attributed to the specific compound and not to a certain impurity, perhaps in the screening sample or whatsoever. So we will make sure that those compounds freshly synthesized hopefully, of course, are responsible for the activity observed in the screen, and you will be provided with the sample, physical sample of those compounds as well. So those of you unfamiliar with the way how we go about let's say, minimizing the number of compounds in the deck, we perform a full deck screen on the maximum of 535 compounds and really take all measures that we can, making the right experiments, deselection, orthogonal assays, target engagement, if possible, to make sure that we select the 50 best compounds. And there we do reconfirmation and resynthesis. So we are really trying to narrow down which compounds from the collection are best meeting your criteria. So general overview, to kind of give you an idea of which partners are involved in the process. The European Screening Center really takes let's say, the initial part of the workflow, review of the proposals, optimization, screening, hit selection, and our medicinal chemistry partners who have also synthesized the 200,000 public compound collection are with us for their synthesis capabilities and their medicinal chemistry expertise. So all these partners together work on your program, on your target program, the screening program, to come up with this QHL list in a comprehensive report, allowing you to progress your results. So a very important topic here is what happens after you've received those results. This is a public-private initiative, and that means that there's always a kind of a balance between the opportunities, the rights, and the obligations. So the program owner has full control over the progression of his or her results. They are provided with a 3 years exclusivity period on the results, so on the compounds on the QHL, for research use and direct exploitation. So you can file patents, you can optimize those compounds, you can publish the compounds if you wish, but there's a 3 years exclusivity period. Within those 3 years, at one point in time, you have to raise your hand to our industry partners saying, hey, industry partners, I've screened this particular target with ELF. I found something interesting. Is any of you interested in collaborating with me to develop this program further along the value chain? Then they are allowed to assess, let's say, the quality of the results. And if they are interested, they can make you an offer for collaboration. You're not obliged to accept that offer, but it's very well maybe a very interesting opportunity for you to license your compounds, to collaborate with them, to have a research collaboration, as we have seen with the Servier case, to further progress your research. In case the program becomes a success, and success is defined as either patent being granted and moving it towards the clinic, so phase 2 clinical trials, phase 3, etc., then there are milestones to be paid to the consortium, and that's only in case of direct exploitation. Clearly, we are very excited to see publications out of the results, but we first like to explore whether there's a direct exploitation potential within your program. So first we go through the so-called option procedure, potential commercial development, and then we move toward publication. Both opportunities are very valid, are possible, and the program owner really directs how to move this forward within the framework that we have just presented. Today we'll be discussing neglected tropical diseases as well, and it's good to mention that the milestone payments are not applicable to neglected tropical diseases programs and malaria. So in summary, the rights and obligations of the program owner, your rights are to have your target screened on the unique compound collection within our screening facility, all expenses paid through the grant that have been given to us, so our hours, use of our infrastructure, the compounds, etc. That's all covered in the grant given to us. If you have a very expensive assay, we may ask you to contribute protein for screening, but that's on a case-by-case discussion. Your rights are to have access to do direct exploitation of the results of the structures of the compounds and full control of the program progression. The obligations which come with public-private funding are, of course, that we need to adhere to the European Commission access rights and dissemination framework. So at one point in time, we would love to see you communicating about the results, either in a scientific publication, on the ELF website, or in any other way. Another obligation, as I just presented, is the compensation to consortium partners, but only in case of direct exploitation. So if this program becomes a success and you move it to clinic, then the milestone payments are to be paid. These payments and milestone payments are transparently listed on the ELF website. They're equal for every program owner, so everyone has exactly the same rules and obligations. So that's very clear and easy to read. The other obligation, as I just presented, is the option for the industry partners to negotiate a license on your program for direct exploitation, to collaboration, license, etc. And that is the obligation to negotiate. You do not need to accept it, but you can see it as an opportunity. So next steps, the application, it's very easy. If you are, after today's webinar, still excited to submit an idea to ELF, you can submit throughout the whole year. We typically have a turnaround time of submission towards, let's say, the news of having your program accepted within 3 to 4 months. We're looking to really recruit a lot of programs within this calendar year. So if you are interested in submitting something, I would say really go through the documentation, reach out to us, and we'll guide you through it. Typically, we'll have eligibility check on the technical details and also, of course, on the program itself. It's been reviewed by the expert panel, and then we'll enter into a legal agreement. The next deadline for submission is September 3rd, so another 2.5 months to work on your application. I would really recommend that you involve your tech transfer early on, because once your program is accepted, we would love to see this signed in an agreement. Some of the French larger research institutes have already signed off on the agreement. For instance, INSERM Transfer is familiar with our legal agreement, other universities and organizations as well, but it's always good to make sure that they are, let's say, early on involved in the process of submitting a proposal. Then the website linked here presents all the relevant documentations and should allow you to submit a proposal. So with that, I'd like to conclude my talk, point you towards the website, and I'm happy to take any questions in the Q&A later once the scientific discussions have taken place. Thank you.
Thanks so much, Jon. I think it's a really great introduction to the European Lead Factory and to what we're doing. In view of time, I suggest if people have any questions for Jon, let's save these, write them down somewhere. We can get back to those during the Q&A session because we have so much other interesting stuff that we still would like to share with you. So I'd like to ask or hand over the floor to Raymond Pierce. As mentioned, he is an Emeritus Director of the Research from the CNRS in France. He will share his experience with the European Lead Factory in high throughput screening of TCDAC2. I hope I pronounced that correctly, Ray, which is a target for the treatment of Chagas disease. Ray, the floor is yours. So,
you have my slides up. My slides are up.
Yeah, they're up, but you still need to put them in full screen mode, if possible.
Yes. Okay. Is that okay?
Not yet, but maybe they're still loading.
Okay. No.
Can you try one more time?
Well, it's I hope I'm on the right one. I'm on the right screen.
Yeah, you're in the right screen, but it's still in, let's say, viewers mode, but not in the...
I'm sorry about that. I'm not sure if I'm... Is that the right one?
I think it's loading. Yeah, now it's correct.
Okay, so now I need to take the control. Ok, bon, merci Vera. Je ferai cette présentation en français et je m'excuse d'avance pour la qualité de cette présentation. Donc comme Vera vous a dit, je suis chercheur émérite au Centre d'infection et immunité de Lille, sur le campus de l'Institut Pasteur de Lille. Et j'ai coordonné, je suis programme owner de ce projet ELF, criblage à haut débit de TCDAC2, une cible pour le traitement de la maladie de Chagas. Et je vais vous partager mes expériences avec tout le processus de cette avec l'OLF. D'abord, je dois préciser que ce projet a vu le jour grâce à un autre projet financé par le CEE appelé Paradise. Nous travaillons sur les maladies négligées parasitaires et nous ciblons particulièrement les enzymes impliquées dans les mécanismes épigénétiques, notamment des effaceurs de marques épigénétiques sur les histones comme les déacetylases ou DAC, histone déacétylase ou plus correctement les lysine déacetylases. Pourquoi? Parce que ces enzymes sont des cibles thérapeutiques pour d'autres pathologies, notamment le cancer, et nous avons pu profiter de l'expérience des différentes personnes avec ces inhibiteurs et les inhibiteurs qui existent comme point de départ pour notre projet. Je vais vous présenter notre schéma classique d'activité pendant le projet. Ceci concerne un autre désacétylase de schistosome Mansoni. Et notre procédé, c'était de produire l'enzyme recombinante qui correspond bien sûr à une cible essentielle pour le parasite concerné, de produire la protéine recombinante purifiée par les méthodes classiques. Nos collègues à l'IGBMC à Strasbourg, dirigés par Christophe Romier, ont cristallisé cette protéine, ils ont résolu ce qui nous a permis deux stratégies de criblage. Un criblage classique en débit, comme pour le ELF, et l'autre, un criblage in silico, que nous avons utilisé également avec la structure de l'approche catalytique bien résolue. Ces deux méthodes nous ont permis d'identifier différents inhibiteurs puissants pour ces enzymes, et nous avons utilisé un système d'optimisation pour optimiser à la fois la sélectivité de ces inhibiteurs, la puissance de ces inhibiteurs pour notre cible, et de voir s'ils étaient actifs contre le parasite, comme c'était le cas pour cet inhibiteur-là. Donc maintenant, je vais parler du projet ELF, le raisonnement pourquoi nous avons dû soumettre ce projet à l'ELF, la construction du projet et les résultats et les conclusions qui en découlent. Nous concernons ici avec la maladie de Chagas. C'est une maladie provoquée par un parasite Trypanosoma cruzi qui est transmis par les réduves et qui sévit dans des pays d'Amérique du Sud, d'Amérique du Central, un petit peu aux États-Unis également. Des millions de personnes sont infectées, il y a des milliers de morts annuels, et la morbidité provoquée par cette maladie aussi représente un fardeau économique pour les pays concernés. Donc, cette maladie procède par différentes phases. Donc, pendant la phase aiguë, après la morsure de la réduve, il y a un peu de mortalité aux phases aiguës, surtout parmi les enfants. Suit une phase indéterminée où le parasite se répand, et vous voyez les formes intracellulaires et le amastigote et les tripomastigotes infectieux. Et cette phase est suivie par la phase chronique pendant laquelle les symptômes graves apparaissent, notamment la cardiomyopathie. Il existe déjà des traitements, benznidazole et nifotimox notamment. Ces traitements sont moyennement efficaces pendant la phase aiguë seulement, pas sur la phase chronique. Et la durée de traitement est très longue. Les effets secondaires sont assez sévères et ceci provoque un problème de suivi de traitement, d'où la nécessité absolue de développer de nouveaux traitements pour cette maladie. Nous avons choisi comme cible TCDAC2, donc c'est une désacétylase, comme j'ai déjà expliqué. Et nos collègues à Curitiba, à l'Institut Carlos Chagas, dirigé par Nissim Zanchin, ont montré que le chaos de cette enzyme chez le parasite provoque un arrêt de la division cellulaire et il n'accumule plus d'ADN. Donc, c'est une cible absolument essentielle pour le parasite. Et de plus, la modélisation de l'approche catalytique montre des différences significatives par rapport aux histones déacetylases humaines. Je n'ai pas précisé pour l'instant que ces enzymes sont bien sûr extrêmement conservées, donc il faut avoir la possibilité d'avoir des inhibiteurs sélectifs. Alors, pourquoi nous avons soutenu ce projet à l'ELF? D'abord parce qu'on avait des difficultés pour la cristallisation de TCDAC2. Donc, cette absence de cristallisation, donc absence de structure définie, rend la stratégie de criblage in silico extrêmement difficile. Nous aurions pu cribler une banque d'un de nos partenaires avec cet hashtag, mais nous avions déjà criblé cette banque avec d'autres enzymes, d'autres lysines d'acétylase. Ces cribles indiquaient l'absence relative d'inhibiteurs de ce genre d'enzymes et souvent générer des dérivés sélectifs. Donc, nous sommes adressés à l'ELF pour avoir la possibilité d'identifier de nouveaux inhibiteurs et surtout, si possible, avec une espace chimique différente dans la banque de l'ELF. Nous avons constitué une équipe parmi les membres du projet Paradise. Donc, j'aimerais profiter pour les remercier de tous leurs efforts. C'est eux qui ont fait tout le travail pour ce projet. Johann Schultz a développé le dosage du substrat. Donc, vous avez déjà précisé qu'il est nécessaire de fournir une méthode de dosage. Christophe Romier et son équipe à l'IGBMC ont produit l'enzyme recombinante pour le projet. Manfred Jung a eu l'idée qu'on devrait se dresser à l'ELF pour ce projet et s'est occupé des dosages enzymatiques suite à la réception de notre QHL, le Qualified Hit List. A vu et resté les composés sur le parasite. Excusez-moi. Notre expérience pendant...
Raymond,
oui.
interrupt,
oui.
but
yeah,
maybe you can switch off your camera because it seems the connection is not too great. And if you switch off the camera, I think it will be less. It will be breaking a little bit less, if that's okay.
Okay, I'll switch off my camera.
Yeah.
Okay.
Thanks.
Okay.
Yeah, this is better.
Désolé tout le monde. Je vais me placer plus près du micro, peut-être. Je ne sais pas si ça change quelque chose. Donc, pendant la soumission du projet, donc, il y a des critères à remplir. Donc, comme ça a déjà été précisé, une nouvelle cible, l'importance du projet au niveau médical et sociétal. Donc, nous n'avions pas de problème à ce niveau-là. La disponibilité de la cible et les méthodes de dosage. Et ça, là, je vais revenir avec les dépôts suivants. Nous avons interagi avec Lygature, avec l'ELF, pendant la phase de soumission et pendant le début du projet, de fois nombreuses par email, par téléphone, et tous ces contacts ont été très précieux et nous ont vraiment aidé dans notre soumission. Je parlerai juste un petit peu des conditions contractuelles. Je suis entièrement d'accord avec le speaker précédent que ça peut poser problème, mais nous avions impliqué notre délégation régionale de l'INSERM dès le départ et ont beaucoup aidé pour expliquer toutes ces conditions et ça ne nous a pas posé problème pour notre projet. Mais il faut dire que notre projet concernait une maladie parasitaire négligée, donc les répercussions financières et les problèmes éventuels au niveau des partenaires industriels sont moins critiques. Post-mission, le délai décisionnel était d'environ 3 mois. Parler un peu de l'enzyme classique. Nous l'avons produit en Escherichia coli, donc c'est encore l'équipe de Christophe Romier qui a fait ça. Purification par les méthodes standards. Là, il y a des exigences de degré de pureté de l'enzyme et il y a donc un certain nombre de cases à cocher. Donc, nous l'avons fourni en solution suffisamment pour 500 000 dosages et un critère très important, c'est sa stabilité après au moins 2 cycles de congélation et de décongélation pendant tout le processus, pour le ligotage, etc. C'est très important. Le dosage de l'enzyme, c'est une méthode qui a été développée aussi par un de nos partenaires, par Johann Schultz, avec un substrat fluorescent et adapté dans nos mains pour 384 puits initialement, pour les formats de 384 puits. Là aussi, il y a toute une série de critères et cases à cocher pour rassurer l'ELF de la viabilité de ce dosage et notamment la possibilité d'utiliser un format de 1536 puits, que le dosage soit homogène, sans lavage, que la lecture soit facile. Dans notre cas, c'est l'intensité de fluorescence, donc ça ne pose pas de problème. Le rapport signal-bruit de fond, toutes ces choses dont vous êtes très familier, je suis sûr. Donc, nous avons également fourni un dossier concernant cette méthodologie pour l'ELF et c'est je crois que ça a aidé à l'acceptation de notre dossier. Je ne vais pas parler du criblage parce que ce n'était pas de notre ressort. 410 000 composés ont été criblés pour ce projet, dont 42 ont été retenus pour le Qualified Hit List après tous les contrôles de faux positifs, etc., après sélection pour les meilleurs inhibiteurs. Après quoi, nous avons reçu les 42 composés et nous avons pu procéder à des tests dans nos mains, notamment Manfred Jung a testé le niveau d'inhibition enzymatique avec d'autres types de dosages, et regarder les structures. Et malheureusement, à ce niveau-là, nous avons trouvé que le niveau d'inhibition que nous avions pour cette cible-là était moyen. Et il n'y avait pas vraiment de nouveauté structurale pour ces inhibiteurs. Donc, là aussi, on était un peu déçus. Il y avait une perspective limitée de sélectivité d'après nos chimistes. Mais nous avons procédé quand même à doser l'activité sur le parasite in vitro. Nielsen Zanchin à Curitiba a utilisé un système d'analyse à haut contenu pour permettre de mesurer l'effet des composés à la fois sur le parasite, ici sont les amastigotes intracellulaires, mais aussi les effets toxiques éventuels sur les cellules. Il a fait 2 séries de dosages, une première série avec tous les composés, qui a permis de sélectionner 8 qui semblent avoir. Et là, dans une deuxième série, je vous montre un type d'analyse de ces résultats, analysés après 48 heures, 72 heures et 96 heures de culture en présence de l'inhibiteur. Ici, ce sont les parasites non traités. Les parasites, le contrôle positif dans nos mains, c'est le benznidazole, c'est l'un des médicaments que j'ai présenté tout à l'heure, qui a une activité qui progresse pendant l'incubation. Aucun des inhibiteurs fournis par l'ELF n'a rempli ces conditions, bien que certains aient été actifs après 48 heures, il n'y avait pas de progression de l'activité pendant la culture. Donc, nous nous sommes arrêtés à ce stade-là. Ce résultat dans nos mains a été négatif, mais je dirais que malgré cela, pour les interactions avec les équipes ELF, mais aussi parce que ça nous a permis de continuer les travaux sur cette cible, bien au-delà du financement européen dont nous bénéficions avec le projet à Paradise. Les techniques et actifs que nous avons développées peuvent être utilisées pour éventuellement cribler d'autres banques. Et maintenant, pendant cette période, Christophe Romier a réalisé la structure cristallographique de TCDAC2. C'est maintenant résolu. Et donc, l'approche par criblage in silico peut procéder. Tout ce qu'il nous faut maintenant, c'est imbécile, mais nous espérons, enfin, mes collègues nous espèrent continuer ce criblage et le développement de médicaments contre cette maladie. Bon, je vous remercie de votre attention et je profite pour remercier les équipes de l'ELF et Lygature pour leur concours pendant tout ce processus et encore mes collègues qui ont fait tout le travail pour ce projet. Je vous remercie.